Good morning, students. Today we'll start the new chapter, that is photosynthesis in higher plants. Photosynthesis is an enzyme regulated anabolic process of manufacturing of organic compounds inside the chlorophyll containing cells from carbon dioxide and water with the help of sunlight as a source of energy. Photosynthesis is a chemical process uses light energy to synthesize organic compounds, that is sugar, carbohydrate. Importance of photosynthesis includes primary source of food and release oxygen to atmosphere. Early experiments. Joseph Priestley in the year 1770 performed a series of experiments with bell jar, candle and mouse. Priestley observed that a candle burning in a closed bell jar soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore to the air whatever breathing animals and burning candles remove. See the pictorial explanation of bell jar experiment. In picture A burning candle inside the bell jar. Soon after, that mouse dies. In third, a plant was kept and then mouse remained alive. That is due to consumption of carbon dioxide by the plants and release of oxygen. The mouse is remained alive. John Ingenhouse in the year 1730 to 1799 showed that sunlight is essential to the plant processes that somehow purifies the air fouled by burning candles or breathing animals. He performed experiment with an aquatic plant and showed that in bright sunlight small bubbles were formed around the green parts while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence, he showed that it is only the green part of the plant that could release oxygen. In the experiment, that hydrilla plant is kept inside the funnel, which is placed in the beaker filled with water. Under the sunlight, that plant performs photosynthesis and releases oxygen, and that is accumulated in the top of the test tube. In the year 1854, Julius von Sas proved that plants produce glucose while growing and stores them as starch. He explained that plants have green substances which is present within a cell in specialized bodies. He found that the green parts in plant is where glucose is made and that the glucose is usually stored as starch. T.W. Engelmann found split light using prism. Vibgyor, green algae cladophora placed in a suspension of aerobic bacteria. Bacteria were used to detect the sites of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. It explained the absorption spectra of chlorophyll A and B. Key feature of photosynthesis was first explained in 19th century, and that plants could use light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organisms was then understood as carbon dioxide plus water in presence of light that forms carbohydrate plus oxygen. Cornelius van Neel. He did experiment with purple and green bacteria and demonstrated photosynthesis is a light-dependent process. He explained hydrogen from H2O reduced carbon dioxide to carbohydrates. He concluded that oxygen comes from water and not from carbon dioxide. Finally, the correct equation for photosynthesis was discovered, and that is 6CO2 plus 12H2O released carbohydrate, that is glucose, 6 molecule of water and 6 molecule of oxygen. Site of photosynthesis. Photosynthesis takes place only in green parts of the plant, mostly in leaves. Within a leaf, photosynthesis occurs in mesophyll cells which contain the chloroplasts. Chloroplasts are the actual sites for photosynthesis. The thylakoids in chloroplast contain most of pigments required for capturing solar energy to initiate photosynthesis. The membrane system, that is grana, of chloroplast is responsible for trapping the light energy and for synthesis of ATP and NADPH. Biosynthetic phase, that is the dark reaction, is carried in stroma of chloroplast where carbon dioxide is reduced to carbohydrate. See the structure of chloroplast. Chloroplast is double membrane bound structure. Outer membrane, inner membrane is there. In the inner space, the membrane bound structure is there, that is known as grana. The unit of grana is thylakoid. And the open space is filled with the matrix, that is known as stroma. In the stroma, ribosome is there. DNA is there, single stranded DNA, that is 70S type. Starch granules are there, lipid droplets are there. Pigments involved in photosynthesis. Chlorophyll A, bright or blue-green in chromatograph. Major pigment act as a reaction center involved in trapping and converting light into chemical energy. Chlorophyll B, that is yellow-green in the chromatograph. Xanthophyll, yellow. Carotenoid, yellow or yellow-orange. In the blue and red region of a spectrum shows higher rate of photosynthesis. What is light reaction? The process which takes place in the presence of light is the light reaction. Light reactions or the photochemical phase includes light absorption, splitting of water, evolution of oxygen, and formation of high energy compound like ATP and NADPH. It involves several complexes, and the pigments of the complex are organized into two discrete photochemical light harvesting complexes. Photosystem 1, that is PS1, and photosystem 2, that is PS2. Light harvesting complex. The light harvesting complexes are made up of hundreds of pigments molecules bound to protein with the photosystem 1 and photosystem 2. The light harvesting complex. Red color, that is the light ray that consists of photons. Photon strikes on pigment molecules, then energy is transferred, an electron is released, and that is accepted by primary acceptor. Each photosystem has all the pigments bound to protein except one molecule of chlorophyll A, forming a light harvesting system, that is antennae. Pigments absorb different wavelengths of lights for efficient photosynthesis. The reaction center chlorophyll A is different in both the photosystems. Photosystem 1, chlorophyll A has an absorption peak of 700 nanometer, that is P700, and photosystem 2, that is PS2, chlorophyll A has absorption peak at 680 nanometer, and that is P680. Process of photosynthesis. It includes two phases, photochemical phase and biosynthetic phase, that is light reaction and dark reaction. Photochemical phase or light reaction, this phase includes light absorption, splitting of water, oxygen release, and formation of ATP and NADPH. Biosynthetic phase, that is a dark reaction, it is light independent phase, synthesis of food material, that is sugar. The electron transport system. In photosystem center, chlorophyll A absorbs 680 nanometer of wavelength of light, red light, causing electrons to become excited and release 2 electrons from the atomic nucleus. These electrons are accepted by primary electron acceptor, that is ferredoxin. The electron from the ferredoxin pass to electron transport system consisting cytochromes. The electron moves in downhill in terms of redox potential by oxidation reduction reactions. Finally, the electron reached photosystem 1. See the pathway of electron transport system. LHC is the light harvesting complex. Light rays, when it strikes on the LHC, electron is released. The electron which is released from LHC, that is accepted by the primary acceptor and then moves to the photosystem 1, the upper one of LHC. And from there again the electron moves and finally accepted by NADPH. Water here splits in presence of light, that is photolysis of water, and electron is transferred to the LHC, because to continue this process there is a need of continuous supply of electron, and that is fulfilled by water. Oxygen is released as a byproduct and 2 proton that combines with NADP to produce NADPH. The electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of photosystem, PS1. The new electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to the another acceptor molecule that has a greater redox potential. Electron hole created in PS1 is filled up by the electron from the PS2. The electron from PS1 pass down hill and reduce NADP into NADPH. During the process of transport of electron, the transfer of electron takes place starting from PS2 uphill to the acceptor down the electron transport chain to PS1. The excitation of electrons transfer to the another acceptor and finally downhill to NADP plus, causing it to be reduced to NADPH. It's called Z scheme due to its characteristic shape, that is a non-cyclic pathway of the phosphorylation, photophosphorylation. This shape is formed when all the carriers are placed in the sequence in a redox potential scale. Splitting of water. Electron required for electron transport system is fulfilled by splitting of water, that is photolysis of water associated with the PS2. PS2 lose electrons continuously filled up by electrons released due to photolysis of water. Water is split into hydrogen, oxygen and electrons in presence of light and manganese ion and chlorine ion. This also creates oxygen to the byproduct of photosynthesis. Photolysis takes place in the vicinity of the PS2. 2H2O released to form 4 proton plus oxygen plus 4 electrons. Photophosphorylation, the process of formation of high energy chemicals ATP and NADPH in the presence of light. Also defined as photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. Photophosphorylation is the process by which the living organism extract energy from oxidizable substances and store this in the form of bond energy like ATP. Photophosphorylation takes place inside the cell in mitochondria and chloroplast. It is of two types, cyclic photophosphorylation and non-cyclic photophosphorylation. Non-cyclic photophosphorylation, two photosystems work in series, first PS2 and then PS1. These two photosystems are connected through an electron transport chain, that is a jet scheme. ATP and NADPH are synthesized by the process. PS1 and PS2 are found in lamellae of grana, hence this process is carried here. Cyclic photophosphorylation, only PS1 works, the electron circulates within the photosystem. It happens in the stroma lamellae, possible location, because in this region PS2 and NADP reduces enzymes are absent, hence only ATP molecules are synthesized, and that is found in bacteria.